It's easy to just open your eyes and see. But when you really stop and think about it, it's this unbelievably complex and, well, frankly, miraculous process. My daughter, Natalie, she was conceived with a single central eye orbit. Just for a little while, thankfully, it corrected itself early on. But it really sparked this fascination in me. Yeah, how does this intricate ability even, develop?
And what's fascinating there, building on that personal connection, is that even something so fundamental, our ability to see, has this incredibly intricate and, yeah, often surprising natural history.
Right.
And that's precisely what we're going to be exploring in this deep dive.
Hmm.
You, our listener, you provided us with this rich collection of material, all under the banner, A Natural History of Seeing. And our mission really is to extract the core insights from everything you shared, from the grand sweep of evolution down to the minute details of how our eyes actually function.
Exactly. So think of this as your personalized journey to becoming, well, exceptionally well-informed without getting totally bogged down in overwhelming detail. We're looking at everything from the origins of vision in the evolutionary tree, the incredible mechanics of the eye itself, how we perceive the vibrant world of color, and even, the ongoing scientific endeavors to restore sight and truly understand what it means to see.
Mm-hmm.
And crucially, we want to understand the eye not as some solitary marvel, but as the source material describes it, as part of a commonwealth of the senses.
That idea, the Commonwealth of the Senses, that's a really vital starting point, I think.
Yeah.
It emphasizes that vision isn't some isolated sort of superior ability, but rather it shares fundamental principles of information gathering, information processing with our other senses, like hearing and touch.
Okay.
Consider Helen Keller. She experienced a sculpture by touch, right?
Yeah.
Meticulously piecing together its form. Now, you might think you see the whole thing in just a single glance.
Okay, yeah, let's unpack this. It's true, I feel like I take it all in seemingly at once, right?
Uh-huh.
But then the source material throws in this really interesting point. It reminds us that our eyes are never truly still. They're constantly making these tiny, rapid, jerky movements called saccades.
Saccades, exactly.
Jumping from one point of interest to another several times a second. And it's between these jumps that your brain pieces together the visual world, like a series of snapshots.
Yeah.
So is my single glance really all that different from Keller's busy fingers exploring the contours, or even like an insect's twitching antenna or a mouse's whiskers?
Well, this raises a really important question about how our brains interpret sensory information, regardless of where it actually comes from.
Right.
And what's fascinating here is the pioneering work of Paul Bachrida. He developed these remarkable prosthetic vests for blind individuals.
I remember reading about that.
Yeah, they converted visual information from a camera into tactile vibrations on their skin. And incredibly, his volunteers didn't just feel shapes, they genuinely began to see, to perceive the forms, even perspective through sensations on their back or stomach. It really seems the brain is primarily concerned with the shape of the information, the pattern, not necessarily the specific sensory pathway it arrives through.
That's absolutely mind-blowing. Light turned into texture, and the brain just figures it out. It powerfully highlights this incredible adaptability, doesn't it?
It really does, the plasticity of our nervous system. And speaking of different pathways leading to similar outcomes, the source material brings up this fascinating concept called convergent evolution. The classic example is the vertebrate eye versus the cephalopod eye, like squid or octopus.
Right. They look similar, but they're not related at all, evolutionarily speaking.
Precisely. Despite being completely unrelated, our eyes develop from specialized skin tissue, theirs form very differently. Both have independently evolved remarkably similar, very efficient structures to achieve vision.
Okay.
This is a powerful example of convergent evolution. Unrelated species independently evolving similar traits to solve the same problem.
So nature kind of finds the best solution and sticks with it, even if it starts from different places.
Pretty much. The key insight here for you is that when a biological problem needs solving, like seeing light, nature isn't endlessly creative with fundamentally different solutions. Instead, efficient designs tend to emerge independently. It showcases the power of natural selection finding optimal pathways. If you need to detect light and form an image, you're likely to end up with something resembling either our eye or, well, a cephalopod's.
Okay, so given this evolutionary push towards vision, how many different kinds of eyes has nature actually come up with? It's clearly not just the single lens model like ours.
Well, the source material points out that most eyes fall into a few broad categories. You've got our single-chambered eyes with a lens, the complex compound eyes of insects and crustaceans, and then more primitive eye spots that can basically just detect light and shadow.
Simple light-dark detectors.
Exactly. But beyond these main types, there's a whole menagerie of truly unusual visual systems out there. Take the tiny crustacean Copelia, for instance. It essentially scans its environment line by line with its constantly moving retinas.
Like a little biological scanner.
Kind of. Creating an image over time. Or consider the brittle star Ophiocoma wenti. Its entire body surface is almost like one giant compound eye.
Wait, its whole body?
Yeah, its surface is covered in thousands of tiny light-sensitive spots connected by a network of nerve bundles.
Wow, a whole surface that sees. That's kind of a wild thought. And when we think about these vastly different eyes, they all have to deal with the fundamental challenge of light itself.
Right.
From blinding sunlight to the faintest glimmer in the dark.
Absolutely. Eyes have to function across an enormous range of light levels. Nocturnal animals like owls and opossums, their pupils can dilate to become almost as large as their entire eye,
Wow.
making them incredibly sensitive in low light, but also quite vulnerable to sudden bright light. Conversely, many animals have evolved really rapid, sophisticated iris adjustments. Think cats with their vertical slit pupils, or geckos with those intricate notched slit pupils. Both can constrict much faster and more dramatically than our human iris can.
And ours. How much do ours adjust?
Our pupils, which are more adapted for daylight, they can dilate maybe about 4 times their constricted width in dim light. That gives us roughly a 16-fold increase in the amount of light gathered. Still impressive, but not as extreme as some nocturnal specialists.
It's amazing how quickly some of those adjustments happen, though. Like that rope analogy the source material uses.
Yeah.
You flick a rope, a wave travels down it. That's light. But then it talks about filtering light, specifically polarizing it, using this analogy of a picket fence.
Yeah, that's a very helpful way to visualize it. Light waves normally vibrate in all directions, right?
Okay.
Polarized light has been filtered, so its waves vibrate mainly in a single plane. So think of trying to shake a rope side to side through a vertical gap in a picket fence the wave won't get through.
Okay.
That's basically how polarizing filters work. They block light waves that aren't aligned with the filter's orientation.
Yeah.
And while we don't naturally sense polarization...
Humans are tool makers.
Exactly. The source material reminds us of that. Like in Raudolf's saga, where Sigurdur apparently uses a sunstone, probably a calcite crystal.
Right. To tell time by figuring out the polarization of sunlight, even when the sun's hidden, that's pretty clever.
Very clever. Using the properties of light in such a practical way. And it's not just about brightness or polarization. Different animals have evolved to see different wavelengths of light too, right? Along the electromagnetic spectrum.
Yeah, like infrared and ultraviolet.
Precisely. Some snakes, certain beetles, they can sense infrared radiation heat, essentially. Helps them find warm-blooded prey in the dark, or maybe find recently burned areas where food might be. George Wald discovered humans can perceive very intense infrared as a dull red glow, but it's not usually relevant for us day to day.
Okay.
Then you have ultraviolet. Many insects, birds, fish, even small mammals like voles can see UV light, which is a shorter wavelength than we can perceive.
What's the use of that?
Well, many flowers have intricate patterns visible only in UV, like little landing strips for pollinators. And voles, which are food for kestrels, they leave urine trails that strongly reflect UV light. Makes them easily detectable for a kestrel hunting from above.
Wow. So it's really interesting how different parts of the light spectrum are important for different animals, like perfectly tailored to their niche. But why don't larger animals generally see far into the UV range? Is there a reason?
The source material suggests a couple of reasons. One, larger eyes gather more light overall, and there's a point where the higher energy of UV light could actually become problematic, maybe overstimulating photoreceptors or even causing damage.
Okay, makes sense.
Also, UV light gets scattered more easily by the atmosphere compared to longer wavelengths. That limits how far it travels, so maybe it's less useful for long-distance vision, which larger animals often rely on.
Right. Okay, now shifting focus a bit. How we perceive the world in 3 dimensions. The source material talks about stereopsis, that sense of depth we get from having 2 eyes.
Yes, stereopsis. It relies on the subtle differences, the disparity between the slightly different images our left and right eyes capture. Our brain then, quite exquisitely, combines these 2 slightly dissimilar views to create a vivid sensation of depth and distance.
And it's mostly primates that have this, right? It's relatively rare.
That's what's particularly fascinating, yes. Stereopsis is relatively rare evolutionarily, primarily found in primates. Many other animals, especially predators like, say, the praying mantis, they rely on a different technique called parallax instead.
Parallax, how does that work?
They'll often rock their heads from side to side or move their bodies and judge the distance based on how the apparent position of their prey shifts against the background.
Okay, so if parallax works, why did primates like us evolve this extra layer of depth perception with stereopsis? The source mentions Jules's theory here.
Yeah, Bill Jules proposed that stereopsis might have offered a significant advantage to early primates navigating complex three-dimensional environments like forests.
Like jumping between branches.
Exactly. The ability to accurately judge distances to branches while leaping, or to precisely locate and grab insects hidden amongst foliage, that would have been greatly enhanced by the fine depth discrimination stereopsis provides.
Makes sense. Okay, so our own bodies are constantly moving, right? Walking, turning our heads. Yet our visual world stays pretty stable. We don't see constant blurring. That stability is largely thanks to things like Purkinje's vestibulo-ocular reflex.
Exactly right. This reflex is triggered by the detection of head movements by tiny sensory hairs within the fluid-filled canals of our inner ear.
The balance system.
Precisely. That information about our head's motion is then rapidly relayed to the muscles controlling our eyes, causing them to move involuntarily in the opposite direction. This counter-rotation perfectly compensates for head movement, ensuring our gaze remains fixed, preventing the world from turning into a blurry mess.
It's truly incredible how many of these essential adjustments just happen automatically, without us even thinking about it. And speaking of things we perceive that aren't necessarily, static, the source material discusses the phi phenomenon, how our brains perceive motion from a rapid sequence of stationary flashes. That's basically how movies and TV work, isn't it?
Precisely. Max Wertheimer, building on earlier observations by Sigmund Exner, he formalized the understanding that if you have two brief, stationary flashes of light appear close enough together in space, and they're presented in rapid succession,
Yeah.
our visual system doesn't see two separate flashes. Instead, it perceives a single object moving smoothly between the two positions. This phi phenomenon, sometimes called apparent motion, is indeed the fundamental illusion behind film and television.
So it's not just persistence of vision like an afterimage.
No, that's a common misconception. Persistence of vision is just the brief afterimage that remains on the retina after a light stimulus is removed. The phi phenomenon is about the brain interpreting discrete stimuli as continuous motion. The source material even points out why sitting in the very front row of a cinema might not be ideal.
Oh, yeah.
The rapid flickering of the individual frames can become more noticeable in your peripheral vision, especially in bright scenes, because your brain has less sort of room to fill in the perceived motion smoothly when the images are that large on your retina.
Huh. Interesting. Okay, so the phi phenomenon creates the illusion of motion, and the vestibulo-ocular reflex keeps our view stable when our head moves. What about when our eyes deliberately track something moving? The source mentions the optokinetic reflex.
Right, the optokinetic reflex is another crucial visual stabilizer. But unlike the vestibulo-ocular reflex, which uses info from the inner ear,
This one uses visual information itself.
exactly. If you're watching a series of moving objects, like stripes scrolling past a window, your eyes will initially track one smoothly. But when your eye reaches the limit of its comfortable movement range, it'll quickly jump back a saccade in the opposite direction to pick up the next object.
Okay.
This rhythmic pattern of smooth pursuit and rapid saccades helps us maintain a stable, coherent view of a moving visual environment.
It's like our visual system has all these complex, built-in stabilization mechanisms working together. But what happens if you manage to completely stabilize an image on the retina, stopping any movement at all? The source describes some fascinating experiments with tiny spotlights on contact lenses.
Ah, yes, those experiments were truly revealing.
Yeah.
When they precisely fixed these tiny spotlights onto contact lenses, so the light always hit the exact same photoreceptor cells regardless of tiny eye movements.
What happened?
The surprising result was that the volunteers actually lost the ability to see the lights after only about a second or two. They just faded away.
Whoa, why?
It dramatically highlights the brain's fundamental need for constantly changing visual input to maintain perception. If an image becomes perfectly static on the retina, the neurons essentially start responding strongly, they adapt, and the image disappears from our awareness.
It's as if our brains just get bored with the same unchanging information and stop paying attention. The source even provides a neat parallel with an AI that couldn't move its artificial eyes.
Right. It learned to achieve a similar effect by moving its entire head in a jerky, saccadic manner, much like a bird does.
That's a really insightful analogy, showing that need for change.
It beautifully underscores the adaptability. Even without typical eye movements, the brain can find alternative strategies to get the changing input it needs.
Now, the source material goes into some really intriguing, almost mysterious areas, like blind sight. That's where someone who's technically blind, due to visual cortex damage, can still react to visual stuff without consciously seeing it, right?
Exactly. The source material describes the compelling case of a patient known as DB. He had damage to his visual cortex, resulting in blindness in a big part of his visual field.
Okay.
Yet, when a stick was briefly held in his blind area while he reported seeing absolutely nothing, he could still guess its orientation, horizontal or vertical, with surprising accuracy. Far better than chance.
So he couldn't see it, but somehow he knew.
It strongly suggests there are unconscious visual processing pathways in the brain that can still receive and analyze visual information, even if it doesn't reach conscious awareness.
It's like there's a whole level of seeing operating below our conscious radar. Really
Yeah.
makes you wonder what else is going on down there. The source then takes this fascinating historical turn, looking at early attempts to understand the eye, starting way back with Galen.
Yes, Galen, the prominent Roman physician, 2nd century AD. He made significant contributions, though his knowledge was limited by restrictions on human dissection then. He famously described the uvea, the pigmented layer, as resembling a peeled grape.
Agree.
Yeah, with translucent blood vessels. Intriguingly, he didn't fully get the retina's function, the actual light-sensing layer. He thought the eye's primary function was maybe related to those blood vessels.
So when did we figure out the retina?
Much later. It wasn't until 1834 that the retina's true role as a, quote, cup of transparent nerve tissue, literally an outpost of the brain, was fully appreciated. So even for such a fundamental part, understanding took centuries.
It's almost humbling, isn't it, how long it took? And the source touches on this rather, well, macabre historical fascination with capturing the last image on a dead person's retina, the optogram. Sounds like something from a Gothic novel.
It certainly does conjure up vivid images, yeah. The idea was that the very last thing a person saw might somehow be imprinted on their retina, maybe retrieved as forensic evidence.
Did it work?
Despite numerous attempts in the 19th century, and that dubious example of the murderer Fritz Angerstein mentioned in the source, generally no. This concept of a persistent last image proved scientifically unfounded. The biochemistry after death just doesn't preserve an image like that.
So no CSI-style reveal of the killer's face on the victim's eyeball. Probably for the best. But speaking of historical understanding, the source highlights the remarkably insightful ancient Egyptian cure for night blindness using liver. That seems surprisingly advanced.
It really is. Night blindness was recognized way back in ancient Egypt, India, China, and the common treatment across these cultures was eating roasted or fried animal liver.
And we now know why that worked.
Absolutely. Liver is exceptionally rich in vitamin A, and as Jordan Wald later showed, vitamin A is crucial for producing rhodopsin, the light-sensitive pigment in our rods, essential for low-light vision. The Ebers Papyrus from around 1550 BC even explicitly describes this treatment.
Wow, but that knowledge got lost.
What's remarkable is that this practical knowledge seems to have been largely forgotten for a long time. Celsus, a Roman encyclopedist writing much later, doesn't mention liver for night blindness. And in the 19th century, a German doctor even wrongly concluded night blindness in orphans was caused by bacteria, completely missing the dietary link.
It almost took a world war to rediscover the importance of diet for vision, didn't it? The source mentions Bloch's experiments in Denmark during WWI.
Yes, the maritime blockades during World War I led to widespread malnutrition. A Danish physician, Bloch, observed a huge increase in xerophthalmia, a severe eye condition from vitamin A deficiency. He did controlled experiments and found that cod liver oil, also rich in vitamin A, was incredibly effective.
So that really nailed the link.
Provided clear, compelling evidence linking vitamin A directly to healthy vision.
And that brings us back to George Wald, whose work the source rightly emphasizes. He really dug into the biochemistry, identifying visual yellow and visual white as forms of vitamin A, figuring out the whole rhodopsin cycle.
Wald's work was truly foundational. He meticulously unraveled the molecular processes in the rod photoreceptors. He showed how rhodopsin is made of a protein, opsin, and a light-absorbing molecule, retinol, which is a form of vitamin A.
And light changes the retinal.
Exactly. A photon of light hits rhodopsin, changes the shape of the retinal molecule, initiating a cascade of events that sends a signal to the brain. In darkness, an enzyme converts retinal back, readying rhodopsin again. Wald beautifully explained how vitamin A is essential for regenerating rhodopsin, and a deficiency directly impairs this cycle, causing night blindness. His work provided the molecular basis for those ancient liver cures.
It's just incredible how these different threads, ancient remedies, wartime observations, molecular biology, all weave together. The source then transitions to the invention of photography with Daguerre. How did that early tech relate to our understanding of the eye?
Well, Daguerre's early photographic processes, while revolutionary, also inadvertently highlighted the eye's remarkable capabilities. His famous 1838 photo of a Paris boulevard needed a really long exposure, 10 to 20 minutes. So while the buildings are clear, the moving people completely disappeared, just blurs. It starkly underscored the eye's inherent ability to capture and process motion in a way early photography just couldn't.
It's like our eyes are constantly taking super fast snapshots and our brain stitches them together. The source also touches on the Cambrian explosion, that period of rapid life diversification, and the role vision might have played, mentioning Parker's in the blink of an eye theory.
Andrew Parker's compelling theory posits that the relatively sudden diversification of animal life during the Cambrian, roughly 540 million years ago, was largely driven by the evolution of sophisticated vision.
An evolutionary arms race triggered by sight.
Kind of, yeah. Better eyesight for predation, better eyesight for avoiding predators. However, as the source points out, the timeline might not perfectly align. The earliest known trilobites with complex eyes appear slightly after the initial Cambrian burst, so vision was likely crucial, but maybe not the single trigger.
Still, a major factor, surely. And the source mentions Nilsson and Pelcher's computer model, showing how relatively quickly a complex camera-like eye could evolve from just a simple flat eye spot.
That's a truly pivotal piece. Their model provided a compelling framework for how such a complex organ could arise through natural selection in a geologically short time, maybe a few 100,000 generations.
How did the model work?
It showed that starting with a simple light-sensitive patch, introducing small, random variations in shape and transparency, and selecting for each beneficial change, a sophisticated camera eye with a lens and retina could indeed evolve relatively rapidly. It helped counter arguments about irreducible complexity.
And some of those early eyes had remarkable features, like the calcite lenses in certain trilobites.
Yes, some trilobites, ancient marine arthropods, had lenses made of calcite, calcium carbonate. Calcite has unusual optical properties. It can refract light for focusing. Intriguingly, in some species, these lenses even had internal layers of magnesium.
Magnesium, why?
Scientists believe it might have helped correct for optical aberrations, reduce blur, suggesting surprisingly sophisticated image formation in these ancient creatures.
Mind-boggling. Okay, the source then draws a clear distinction between the camera-type eye, like ours, and the compound eye of insects and crustaceans. What are the key differences and trade-offs?
Right. The camera eye, like ours, has a single lens focusing light onto a retina, creating a detailed image. It likely evolved from a light-sensitive patch folding into a cup, developing a pinhole, then a gel-like substance, and finally a proper lens.
And compound eyes.
Compound eyes are built from numerous individual units called ommatidia. Each captures a tiny sliver of the scene. The brain then integrates signals from all these ommatidia into a mosaic-like image.
So a camera eye gives higher resolution.
Potentially, yes, but having numerous independent camera eyes would be energetically very expensive to build and maintain.
So, if compound eyes typically give what the source calls a rough, pixelated view, and Nilsson even said evolution seems to be fighting a desperate battle to improve them, why are they still so common? What are the advantages?
Despite lower resolution, each ommatidium is like one pixel. Compound eyes offer significant advantages, especially for smaller animals.
Like what?
Firstly, size. Having large bulbous eyes isn't as much of a physical burden for small creatures. Secondly, and perhaps more importantly, they excel at detecting movement.
Better than ours.
In some ways, yes. Our high resolution can actually introduce motion blur with fast-moving objects. Compound eyes, with their lower resolution and arrangement, are incredibly sensitive to even slight movements, crucial for detecting predators or tracking fast prey. Think of a wasp navigating rapidly.
Right, okay, so great motion detection. The source also mentions predatory insects using silhouette vision. How does that work?
Exactly. Because detailed images against complex backgrounds are tricky, many predatory insects use a clever strategy. They position themselves below their prey and look up.
Ah, so the prey is silhouetted against the bright sky.
Precisely. This high contrast view makes even indistinct shapes stand out clearly, making detection and tracking much easier.
Smart. And compound vision is important for flight control, too. The source mentions Grinley's work with pilots and bees using optic flow for landing.
Yes, G.C. Grinley explored how both pilots and flying insects use optic flow, the apparent movement of the visual scene as you move through it, to gauge speed, altitude, distance, especially during landing.
How does a bee use it?
As a bee approaches a flower, the visual textures of petals and leaves appear to move faster across its compound eyes. The bee's brain interprets this increasing optic flow as getting closer, and it adjusts its flight speed for a smooth landing.
Astonishing how much info they get from simple input. And as we touched on, some trilobite eyes, like Cyclopes, had those magnesium layers in their calcite lenses. What does that imply?
The magnesium layers suggest their eyes weren't just simple light detectors, but could form relatively clear, focused images. The different refractive indices likely corrected for optical aberrations. This optical sophistication implies a more complex nervous system capable of processing these details, suggesting a more active, visually guided lifestyle than we might assume for such ancient creatures.
So even within compound eyes, there's huge variation. The source also mentions superposition, compound eyes for low light, and Kirschfeld's discovery of overlapping visual fields in flies.
Right, superposition eyes, found in nocturnal insects, have multiple ommatidia focusing light onto a single photoreceptor, increasing sensitivity at the cost of some resolution. Kirschfeld's work on flies showed their ommatidia have slightly overlapping fields of view, and the neural processing combines these signals, effectively sampling each point multiple times. This neural oversampling enhances faint light detection.
Okay, moving to the vertebrate eye, our focus. The source discusses inherent optical imperfections like spherical and chromatic aberration. Can you break those down?
Sure. Spherical aberration happens in spherical lenses because light rays hitting the edges bend more strongly than rays hitting the center, so they don't all focus perfectly, causing slight blur. The source notes land vertebrates generally evolved flatter lenses than aquatic ones, which helps reduce this.
And chromatic aberration.
That occurs because different wavelengths or colors of light bend by different amounts when passing through a lens. So different colors focus at slightly different points, again reducing sharpness, sometimes causing color fringes.
How do eyes deal with that?
Vertebrates have various strategies. Squirrels have corrugated retinas, maybe to put photoreceptors at different depths. In humans, blue-sensitive cones are less concentrated in the central fovea, our sharpest vision area,
Why?
because blue light tends to focus slightly in front of the retina. Having fewer blue cones centrally minimizes its impact on sharp vision. This might also be why blues and greens can seem to recede in paintings.
Fascinating. Evolutionary workarounds for imperfect optics. The source then shifts to the crucial role of movement and attention, starting with Lorenz's classic studies on imprinting in birds.
Konrad Lorenz famously showed imprinting, where newly hatched birds like geese form a strong attachment to the first moving object they see, usually the mother, but Lorenz showed they could imprint on him, or even inanimate objects. It powerfully highlights the role of early visual experience in shaping innate behaviors.
And it's clear our brains don't just passively record, they actively interpret and make assumptions, right?
Yeah.
Lorenz mistaking a distant mill for a steamer ship illustrates that.
Exactly. Our brains are efficient, relying heavily on prior experience and expectations. We don't meticulously process every detail. We make rapid subconscious inferences. Lorenz's momentary mistake is a perfect example. The source also mentions Emma Kay trying to write the Bible from memory.
Right, showing how fallible recall is, how our brains fill gaps plausibly but not always accurately. It's almost like we see what we expect to see. The source also mentions the surprising power of simple eye-like shapes, dots in a circle, to trigger recognition. Seems very fundamental.
It is indeed. Evolutionarily, quickly detecting eye's predator, prey, social partner is crucial for survival. Even a basic configuration like 2 dark spots in a lighter area can trigger a rapid, unconscious, attentional response in many animals, including us. It reflects the deep evolutionary importance of assessing gaze and intentions.
And the source then goes into Chance's experiments with rats, the stress of having nowhere to hide, and connects that to humans using unpredictability in social interactions. That's a bit of a leap.
It's an intriguing parallel. Chance found rats denied hiding places showed significant stress. He then suggested that in primate social hierarchies, unpredictable behavior by dominant individuals might create a sort of perpetual terror for subordinates.
Removing the hiding place of knowing what to expect.
Kind of, yeah, leading to chronic stress and reinforcing dominance. It highlights the complex, often subtle dynamics in primate social structures.
Unsettling, but interesting. And a key element there is gaze where eyes are looking. The source discusses our innate tendency to seek and follow gaze, starting really early in babies.
Yes, research shows from as early as 2 months, infants prefer looking at eyes. By 6 months, they spend more time looking at faces, gazing directly at them. This develops into following another's gaze, fundamental for shared attention and later, language acquisition. Progressing from glancing to pointing is a key milestone. Similar gaze following is seen in baboons, too.
We seem hardwired to connect via eyes. And the source also touches on sensitivity to facial expressions, noting how inverted faces look monstrous. Why is that?
Our brains are highly specialized for processing faces holistically as a configuration. When a face is inverted, this holistic processing is disrupted. We become more aware of individual features in an unusual orientation, which feels unsettling, even grotesque. It underscores the sophisticated configural nature of facial recognition.
Which leads to Paul Ekman's work on universal facial expressions and the debate, innate or learned.
Ekman's cross-cultural research provided strong evidence for the universality of certain basic expressions linked to emotions like happiness, sadness, anger, fear, surprise, disgust. While some argued expressions were culturally learned, Ekman's studies, showing consistent recognition across diverse cultures, strongly support the idea these core expressions are largely innate, biologically determined. Darwin explored this too, actually.
But the face communicates more than just raw emotion, right? Meaning intent, which leads to deception, like false smiles.
Exactly. We learn to consciously control facial expressions to communicate specific meanings or intentions, which can include concealing true emotions. The false smile is a common example of using facial muscles to convey a social message not matching our genuine state. Signaling enjoyment isn't always the smile's most critical function in all social contexts, evolutionarily speaking.
Okay, let's delve into the history of early theories about vision and light. The source starts with Theon of Alexandria, 4th century AD, viewing the eye as an active hunter, not passive. Seems quite dynamic for the time.
Theon's analogy is insightful. Comparing the eye to a hunter emphasized its active role in exploring the world, unlike the passive reception of sound by the ear, a significant departure from purely passive models.
Then we get extramission. The idea visual rays come from the eye. Plato was big on this.
Yes, Plato incorporated extramission. He theorized our eyes emit subtle visual rays that actively reach out and interact with the forms or essences of objects, enabling perception. It was one way to explain perceiving things we haven't encountered before.
Quite different from light entering the eye. Aristotle, Plato's student, pushed that intromission theory, but struggled to explain focus, right?
Aristotle's intromission theory, something travels from the object to the eye, is closer to our modern view. But without understanding optics, he struggled to explain focusing at different distances, why vision wasn't just a blur.
Then Euclid approached it geometrically with his optica.
Euclid, around 300 BC, focused on the geometry of light rays traveling in straight lines, angles explaining size, distance, shape. But his theory was primarily geometric, lacking biological detail about the eye or subjective experience.
Fast forward several centuries to Al-Kindi, who tried to reconcile physics, geometry, psychology, but still favored extramission, and used reading as an example.
Al-Kindi tried integrating disciplines. His argument for extramission partly relied on observing we see clearest at the center of gaze. He reasoned if the eye were just a passive receiver, why actively scan text when reading? However, his ray theory had a flaw. Why wouldn't returning rays scatter and blur the image?
A big hurdle. Then Alhazen, or Alhazen, made a much stronger case for intromission, seeing the eye as a camera obscura. But he struggled with the inverted image problem.
Alhazen was pivotal. He fully embraced intromission, provided evidence, accurately described the eye as a camera obscura forming an inverted image on the retina. But without understanding the nervous system and brain processing, he couldn't resolve the paradox. How does an inverted retinal image lead to upright perception? The idea of perceiving an inverted world seemed nonsensical. Bridging optics and subjective experience was the challenge. Nerve signals were still largely unknown.
Really highlights how understanding the optics had to come first. It was Kepler, the astronomer, who gave us our current basic model of the eye forming an image, almost as a side project.
That's right. Kepler's 1604 work provided the essential foundation. He correctly described how the lens focuses light onto the retina, forming a real inverted image. Pivotal insights, even if astronomy was his main focus.
And around the same time, Felix Platter made crucial anatomical observations, identifying the lens and its attachments. Seems like pieces were falling into place.
Platter's detailed anatomical work in the late 16th century provided essential biological details complementing optical theories. Correctly identifying the lens and ligaments furthered understanding beyond abstract optics, helping solidify intromission.
For a surprisingly long time, people believed bright eyes generated their own light. Right. Seems intuitive, but wrong.
It was a common belief for centuries. Luminous eyes emitting light seemed a simple explanation. Even in the 17th century, some claimed they could read by their own eyelight. Observing reflective surfaces in animal eyes, fish scales, the tapetum lucidum in nocturnal animals like horses likely contributed to this idea.
But it was Rudolphi, Purkinje's student, using a decapitated cat, who showed the reflection was just physics, not biology. And this led to studying the living eye via reflected light.
Rudolphi's experiments, though unsettling, were significant. Demonstrating light reflected from a cat's eye traveled the same path as incoming light provided strong evidence eye shine was passive reflection, not active generation. This realization paved the way for instruments to illuminate and view the eye's interior by observing this reflected light, leading eventually to the ophthalmoscope.
Which brings us to Charles Babbage, the computer pioneer, and his early ophthalmoscope attempt. But it wasn't a big hit initially. Even Wharton Jones wasn't impressed.
Babbage's early ophthalmoscope, conceptually groundbreaking, had practical limits. Light emerging from the eye is slightly convergent. His design lacked a diverging lens for the observer, so Jones likely struggled to get a clear focus. A simple diverging lens could have significantly improved it, maybe leading to earlier adoption.
Ironic how a small design oversight can hinder a breakthrough, but refining the ophthalmoscope was crucial, allowing direct retina viewing, discovering the blind spot where the optic nerve exits, no photoreceptors, and how our brain fills it in. Another clever brain trick.
Absolutely. Viewing the retina directly was monumental. Discovering the blind spot, realizing our brain actively interpolates missing info based on surroundings, beautifully illustrates perception's constructive nature. We don't see a black hole because our brain diligently fills the gap. You can easily find your own blind spot.
Astounding how much the brain does behind the scenes. The source then discusses the retina's intricate structure. Miller discovering distinct nerve layers, Schultz's detailed drawing showing 10 neural layers. Sounds incredibly complex.
It truly is. Mueller, identifying the layered structure initially met with skepticism, was pivotal. Schultz's meticulous work, using new techniques, revealed incredible cellular complexity photoreceptors, rods, cones, bipolar cells, ganglion cells, interneurons, all highly organized. This intricate Heath-Robinson arrangement, as the source jokes, immediately raised the question: how does this circuitry process light signals into meaningful information? Still an active research area.
And Purkinje, mentioned earlier for the reflex, also made early observations of subjective visual phenomena, like the Purkinje shift in color perception in different light.
Yes, Purkinje was a keen observer of his own vision. The Purkinje shift, how relative brightness of colors changes as light dims, blues-greens seem relatively brighter than reds, was one contribution showing how perception changes, reflecting differential rod cone sensitivity.
It seems counterintuitive that the retina is back to front, with photoreceptors at the back, blood vessels and nerves in front, blocking light. Why isn't that a huge disadvantage?
While it seems suboptimal, the source points out advantages. Blood vessels in inner layers provide vital nutrients oxygen to highly active retinal cells. And we don't typically perceive these vessels. They're close to photoreceptors, and our visual system adapts, making them largely invisible.
What about the fovea, our sharpest vision spot?
In the primate fovea, this issue is addressed more directly. Other retinal layers and blood vessels are displaced aside, creating a clear path for light to reach densely packed cones, ensuring optimal resolution. The choroid layer behind the retina also nourishes photoreceptors there.
So functional reasons for the seemingly inverted setup. The source then transitions to the neuron doctrine and its impact, highlighting Ramon y Cajal using Golgi's stain to show the nervous system is individual cells' neurons, not a continuous network.
Ramon y Cajal's meticulous work using Golgi stain provided compelling evidence the nervous system, including the retina's circuitry, isn't a continuous reticulum, but countless individual neurons communicating at synapses. This neuron doctrine was a paradigm shift, providing the cellular framework for understanding how sensory info, including vision, is processed.
And this paved the way for Hartline and Graham studying horseshoe crab nerves, finding light intensity encoded by firing rate, and Hartline discovering lateral inhibition.
Hartline and Graham's studies on horseshoe crab photoreceptor nerves were fundamental. They showed stimulus intensity translates directly into the nerve fiber firing frequency. Hartline also discovered lateral inhibition. An active photoreceptor inhibits its neighbors.
What does that do?
It enhances contrast, sharpens edge perception, making shapes easier to discern. A lot of processing happens right in the retina before signals even reach the brain.
So that helps explain why we're so sensitive to local contrast, inferring shading from images like Dawkins' Che Guevara contrast-only picture. Our brain actively constructs reality.
Precisely. We're far more sensitive to changes in intensity contrast than absolute levels. Our brain relies heavily on these local differences to infer shape, form, shading. Dawkins' image dramatically shows how much the brain fills in based on edges.
And the distribution of rods and cones reflects this trade-off between sensitivity and acuity, or detail.
Yes. Rods, mostly in the periphery, are highly sensitive to low light, night vision, but don't give sharp detail or color. Cones, concentrated in the central fovea, give high acuity color vision in brighter light. Their receptive fields, how many photoreceptors feed one output neuron, also differ. Foveal cones have small fields for fine detail. Peripheral rods have large fields for sensitivity, sacrificing acuity.
Okay, let's turn to color perception. Fascinating, oft-debated. Edwin Land's Retinex theory proposed color is more subjective, dependent on neural processing.
Land's Retinex theory challenged the view that color is solely determined by wavelengths hitting the three cone types. Based on experiments with colored Mondrian patterns under different lights, he argued color perception is more complex, involving sophisticated neural computations.
How so?
He proposed our brain constantly compares relative reflectances of surfaces across different wavelengths in a scene. It's these relative comparisons, not absolute wavelength, that determine perceived color.
And even language can influence color perception. Davidoff's work with the Barinmo tribe in New Guinea.
Davidoff's research was fascinating. The Brunello language lacks a distinct term separating blue and green. He found while they could physically discriminate hues, they struggled to remember subtle differences between shades of green after a delay unless they had a specific term for those shades. Suggests the verbal labels can shape how we perceive and remember color. Language, cognition, perception are intertwined.
Makes you reconsider Gladstone's controversial idea that ancient Greeks had different color vision based on Homer's odd color descriptions, like the wine-dark sea.
Gladstone's theory remains debated. His argument, based on Homer's unusual terms, wine, dark sea, green honey, proposed Greeks might have had a less nuanced color experience, maybe reflecting earlier evolution or different categorization. Hard to prove, but prompts thought about cultural linguistic influence.
The source also mentions how environment shapes color experience, like Finland's intense red-pink sunsets due to atmospheric scattering at high latitudes. Shows it's not just the eye, but light interacting with surroundings.
Absolutely. Unique interplay of sunlight and atmosphere creates diverse visual experiences. Long, shallow sunlight path at high latitudes scatters shorter wavelengths, blue, green, leaving longer red surges dominant at sunset, sunrise. A beautiful reminder color perception is linked to light physics and environment.
And on a human note, the explosion of synthetic pigments in the 19th century must have dramatically expanded the colors people experience daily.
Truly revolutionized the visual world. Before, available colors were often limited, expensive. New synthetic dye pigments brought vibrant, stable colors to the masses, profoundly influencing visual culture, fashion, art. Undoubtedly broadened color experiences.
Back to biology. Thomas Young's three-receptor theory in the early 19th century was foundational, right? Even if later refined.
Young's hypothesis was a landmark. He proposed our eyes have three receptor types, each maximally sensitive to different wavelengths, initially red, yellow, blue, later red, green, violet. Based mainly on color mixing observations, combining these three primaries creates a vast spectrum, a compelling, simple explanation for our rich color perception.
And his trichromatic theory explains complementary colors and afterimages related to cone fatigue.
Yes, complementary colors, pairs mixing to white-gray, and afterimages fit well. Stare at a strong color, cones sensitive to those wavelengths fatigue. Look at white, less fatigued cones respond more strongly, giving an afterimage in the complementary color, for example, red stimulus, green afterimage. Helmholtz further developed Young's ideas into the widely accepted Young-Helmholtz theory.
So there was young Helmholtz, but then Ewald Hering proposed his opponent process theory. Opposing pairs like red-green, blue-yellow. Sounds like a big debate for a while.
Indeed, a lively, sometimes contentious debate through the late 19th century. Hering's theory was based on psychophysical observations. Certain color combos, reddish-green, don't occur, complementary afterimages. Trichromatic theory explained color mixing well, but struggled with these phenomena.
So who's right?
Much later, it emerged both are correct, but at different levels. We have trichromatic reception in cones, young Helmholtz, but signals are then reorganized into opponent channels, red, green, blue, yellow, by neurons in the retina and thalamus, Hering. Complementary pieces of a larger puzzle.
Fascinating. The source also mentions James Clerk Maxwell's color mixing experiments using filters and magic lanterns, providing strong empirical support for trichromacy.
Maxwell's meticulous quantitative experiments in the mid-19th century were compelling evidence, projecting overlapping red, green, blue beams showing he could match any color by adjusting intensities, provided strong experimental backing for Young's theory. His use of filters and magic lanterns was groundbreaking.
And Land's-Redneck's theory, well, different. Built on this earlier work, but really shifted focus to the brain's active role in constructing color experience. The source also mentions flicker colors, like Benham's top black-white patterns evoking faint colors. Still mysterious.
Yes, Benham's top and other flicker-induced colors remain an active research area. Suggests our visual system can generate weak color sensations without specific color wavelengths in the input, further highlighting perception's complex constructive nature, the interplay of temporal changes and color perception.
The history of reproducing color includes early printing attempts, like Leblon in the 18th century, using 3 primaries plus black-white. Amazing how early color-mixing principles were applied practically.
Leblon's early 1700s work was significant. He recognized most colors could be reproduced mixing 3 primaries. He used red, yellow, blue, plus black-white for depth tone. Rudimentary techniques, but his conceptual contribution was influential.
Culminating artistically in pointillism Seurat, Signac placing pure color dots, relying on the viewer's eye to blend them optically. Art applying vision science directly.
Absolutely. Pointillism was a direct application of 19th century color perception understanding. Seurat and Signac consciously used optical mixing. Small, unmixed color dots viewed from a distance blend in the viewer's visual system, achieved unique luminosity and vibrancy.
Colorblindness also offered insights. The source mentions the Lagerlunde train crash, though not caused by it, raising awareness, leading to tests like Thompson's. And Dalton, studying his own colorblindness, was pivotal.
Recognizing colorblindness and its potential safety implications did lead to systematic testing. While the Lagerlunde crash wasn't due to it, it drew attention. John Dalton's detailed description of his own red-green color blindness, initially wrongly attributed to blue eye fluid, was crucial. His observations were later explained by Young's theory missing or malfunctioning red or green cone pigment.
The genetics of color vision solidified understanding, especially with Mendel's laws rediscovered, chromosome theory, and then Jeremy Nathan's identifying the specific genes for red, green, blue receptors on specific chromosomes.
Nathan's late 1980s research provided definitive molecular evidence for trichromacy and explained colorblindness inheritance. He identified and cloned the R, G, B pigment genes, mapped them. Red and green genes are close together on the X chromosome.
Explaining why red-green color blindness is more common in males.
Exactly. Males have 1X, females 2. Their proximity also makes them prone to recombination errors, causing various RG deficiencies. The blue gene is on a different chromosome, hence blue color blindness is rarer, inherited differently. Nathan's even suggested some women inheriting different RG versions on their 2Xs might have slightly enhanced color discrimination.
Incredible path from color mixing observations to specific genes. The source also highlights Gordon Wall's understanding photoreceptor evolution, how visual systems adapted, noting early mammals throwing away sophisticated vision due to nocturnal life.
Wall's provided a comprehensive evolutionary perspective, emphasized adaptations are linked to environment and behavior. Early mammals, evolving when dinosaurs dominated, were largely nocturnal, lost some complex color vision, relied more on rods. Full trichromacy re-evolved later in primates. Walls also revisited chromatic aberration and blue cones outside the fovea as evolutionary compromises.
And Nathan's genetics support independent evolution of trichromacy in Old World monkey apes like us, versus dichromacy in most other mammals. And neural processing in the LGN supports Hering's opponent theory.
Nathan's studies strongly suggest trichromacy evolved relatively recently in primates. Red-green genes likely arose from duplicating an ancestral green gene. Old World monkey apes have 3 functional pigments, most mammals have 2, blue-green, resulting in dichromacy. And yes, opponent processing in retinal ganglion cells and the LGN thalamus organizing signals into red, green, blue, yellow channels provides clear physiological support for Hering's theory at a neural level beyond photoreceptors.
The source also touches on acquired achromatopsia losing color vision due to brain damage, and Zicky's findings suggesting some unconscious color processing might persist. Shows the system's complexity and redundancy.
Yes, acquired achromatopsia cases offer further insights. Zicky's research suggests while conscious color perception is lost, some unconscious processing might occur elsewhere, highlighting color processing's distributed, multifaceted nature in the brain.
The final section looks ahead. The historical quest for mechanical mind analogies, Mach needing a new sensation vocabulary. Then ethically complex areas like non-lethal visual weapons, dazzling lasers, and controlling vision.
The tendency for mechanical analogies reflects our ongoing effort to understand consciousness. Mach rightly saw their limits for subjective experience, advocating a new vocabulary. Non-lethal visual weapons raise profound ethical questions about misuse, consequences. Manipulating vision carries significant ethical weight, increasingly critical as tech advances.
Then attempts to restore sight. Early cataract surgery, Cheselden, Maria von Paradis, Mesmer, highlighting the role of movement in learning to see after blindness. And Ames' illusions, Ames' room, showing perception, relies on assumptions, guesswork.
Early sight restoration attempts revealed correcting optics isn't enough. Patients struggled interpreting info, highlighting the critical role of early visual experience and active learning through movement touch. Cases like Maria von Paradis and Cheselden's patient illustrate the brain's challenge with new visual input. Ames' illusions underscore perception isn't direct recording, but active construction based on knowledge assumptions.
This movement vision link inspired robot eyes, drawing from insect vision for navigation, optic flow airspeed, polarized light navigation. And COG aimed to create robots learning via vision and touch, like infants.
Biological vision principles, especially insect eye efficiency for surveillance motion detection, have inspired robotic vision. Robots use optic flow for navigation, estimating airspeed. The ambitious COG project aimed for a humanoid robot learning via multimodal senses, integrating vision touch like human development.
And finally, more direct attempts. Bypassing eyes to stimulate visual cortex. Brindley, Dobell electrode implants. Huge potential, but technical hurdles, ethical issues, volunteer death. Also, artificial retinas stimulating remaining retinal layers, and augmented reality, AR, overlaying digital info, concluding with change blindness, Grimes, and inattentional blindness, Door study, showing how much we miss.
Direct cortex stimulation, phosphenes, was bold but faced implant stability resolution issues and ethical challenges. Artificial retinal prostheses offer a less invasive approach for degenerative diseases. AR represents a different way to augment vision. And yes, change intentional blindness are powerful reminders. Perception isn't complete recording. We miss significant things if attention is focused elsewhere, highlighting visual awareness's selective nature.
So to bring it all together, this deep dive shows vision as incredibly complex, multifaceted, shaped by evolution, intertwined with biology, environment, even social interactions. It's truly not passive light reception, but a remarkably active interpretive system.
That's a crucial and concise summary, yeah. Vision is active, constructive, shaped by evolution, encoded in genes, realized through intricate neural circuits, constantly influenced by attention and expectations. A testament to natural selection sculpting such a vital system.
And it really makes you wonder, doesn't it, given how much our brains filter and interpret, how much we can miss even when looking right at something. What other fascinating aspects of our reality might be going unnoticed right under our noses? Perhaps gaining this deeper understanding of how vision works can even subtly shift how we perceive our own daily experiences and the world around us.
That's a wonderfully provocative thought to leave you with. The world of vision is indeed full of surprises, and our own perception is far less complete, far more interpretive than we often realize.
Thank you for embarking on this deep dive with us into the extraordinary natural history of seeing. We hope this exploration has illuminated some of the incredible intricacies of this fundamental sense and perhaps even changed the way you see the world just a little bit.